excited. Thank you so much. And it's really a pleasure to be here. In addition to the fact that I published a paper first with Alex in 1992, I joked that we were, of course, in middle school at the time. It's been incredible to really see the work that's come out of this institution and over the years, and so many different viruses, but most recently the really substantive work in SARS-CoV-2 and the pandemic. So really a pleasure to be here. I don't have any relevant disclosures. And so I'm going to start really with something that we published, and I might skim through bits of it in part because it is published. So we basically were really wondering, why is it that some people become so severely ill during the acute phase of COVID, which of course many people have wondered, and we still don't totally understand. And others, progress to serious illness and death, while some recover and do just fine. And for comparisons, we actually, of course, looked at their responses compared to just what's going on the average day with a healthy person. We looked at acute hepatitis C because of early evidence of inflammasome activation in SARS-CoV-2. My lab showed that hepatitis C really robustly induces the inflammasome in the acute phase. So that would be a good comparison for, what happens with changes with induction of the inflammasome, that innate sensing and inflammatory pathway. Chronic hepatitis C just as a comparator for the hep C. And then because it's a respiratory pathogen, really what happens in patients who are hospitalized with influenza. And we focused this was the work of Liz Thompson, who was working with Jonathan Powell and with me, and Katie Casino, a grad student in my lab, and was published. And we divided really into T cell as well as myeloid and B cell panels. And these are very broad panels that allowed us to look at many of the different types of the immune cells that we care about, 36 different subsets. But we said, well, okay, if, the analogy is I'm standing at the finish line of a race and I look at everybody who's standing there, we can count up all the people of different types, just like we count immune cells that are present. But probably the easiest way to tell at the end of the race who ran the race and who's standing there cheering is to look at their metabolism. So we said, let's not just look at what cells are present, but what is going on with them metabolically so that we could ideally identify those that are most actively participating in this process. And so that really comes down to work that's been honed further since that initial publication by Liz Thompson, again with this T cell panel and the myeloid and B cell panel, but with the addition of metabolic markers that help us to figure out what is happening in the cells that we identify. So probably most importantly to focus on are a few of these things. CPT1A basically is the rate-limiting enzyme in long-chain fatty acid oxidation. GLUT1 is a glucose transporter and key in glycolysis, hexokinase 2, and I'll show a little map in a second for people who are having PTSD and thinking about when you had to learn about metabolism in earlier phases of your career. And then TOM20, or translocase of the outer mitochondrial membrane complex, subunit 20, is critical mitochondrial generation of OXPHOS, as is VDAC, or voltage-dependent anion channel. So again, as just a little bit of a refresher, at baseline, many cells, for example, resting T cells, have just kind of low-level oxidative phosphorylation going on. But when they become activated, basically they upregulate oxidative phosphorylation and fatty acid oxidation, but they also begin to use glycolytic pathways. And again, I've pointed with arrows to the places that things that we've used to assess whether glycolysis is going on. And as cells become resting memory cells, they decrease that some. And maybe a little bit misleading here because I've said activated macrophages and dendritic cells use glycolysis, particularly M1 macrophages use glycolysis, so it's not universally true of all the subsets. But at any rate, we are able to actually take a look at T cells and say, are they, and this is intracellular cytokine staining, are they actually undergoing glycolysis? And that would be measured by their hexokinase 2 levels and GLUT1 glucose receptor levels. And we ask, whether this is, there's evidence that glycolysis is going on, or in the absence of those molecules being upregulated, perhaps a more oxidative phosphorylation pattern. And again, this is a bit of a simplification, and I'm conscious that, people like Rachel know quite a bit about this. So, but basically we then said, well, how can we really assess, oxidative phosphorylation or fatty acid oxidation? And we used both TOM20 and VDAC1 for that, as mentioned. So one of the things that we noticed was that, there is a really massive upregulation of VDAC on the surface of both CD4 and CD8 T cells, and that is compared to healthy controls. But again, we wanted to say, well, okay, obviously it's not hard for somebody to tell the difference between somebody with severe COVID and who's running around totally fine. So let's be a little bit more logical in our comparisons here. And in fact, this is also present in acute influenza, but not in acute HCV, so clearly not strictly driven by inflammasome activation, for example. And this actually tends to dissipate as people recover. So these are in the acute phase of COVID and in recovery, and we find that there's actually a decrease in the presence of this population of cells. So again, not really telling us why this looks different than severe influenza, but if we actually compare these T cells that are, again, VDAC high, and look at individuals with flu, they're not the same. So basically, in the acute phase of SARS-CoV-2, we see these cells segregating, except for there's always got to be one person who just misbehaves. But and then the recovered individuals, and here are the people who were acutely, severely ill with influenza. And in fact, when you look at these T cells, while you see high levels of VDAC, the cells are coded in the histogram according to the labels here. So these dark blue are the cells from the people with COVID, and they have lower levels of GLUT1, indicative of less glycolysis and less T cell effector memory activation, and then more TOM20, again, indicative of mitochondrial membrane dysfunction, and higher levels of KLRG1. So actually, although they started out looking similar with VDAC, they actually have distinct profiles, and particularly in those with severe disease. So again, on this pathway of activation, T cells tend to use less glycolysis until they're activated, and then when they become exhausted, you see similarly less use of this. So the question was, what is going on in these cells? One of the things, and again, this is a highlight because this is published papers, is we actually then did some single-cell RNA-seq and found of people who had more than 50% of their cells, their T cells, these VDAC high positive cells, and found that they had evidence of cell death pathways being activated and decreased pathways involved in normal mitochondrial function. And so when Ann Hamaker-Brady did some EM images for us and showed basically that their mitochondria are really dysmorphic as well. So these are the patients with COVID, and instead of having these nice mitochondria that we all learned about with these well-formed cristae, they're basically very dysmorphic. And that is indicative of cell death. And an important point is that we could also do confocal microscopy to look at the presence of mitochondrial abnormalities. And an important point here is that VDAC overexpression and oligomerization has been associated with apoptotic signaling. So the question is, do we have cellular evidence of apoptosis going on? And the answer is yes. So the normal pattern of cytochrome C, which is released broadly throughout the cell when cells undergo apoptosis, is the normal pattern is it's actually restricted to a very narrow range in the cell and then becomes released and is present diffusely throughout the cytochrome in cells undergoing apoptosis. And you can see that both on staining here, and this is a healthy control where you have this restricted localization of cytochrome C as opposed to patients with COVID where cytochrome C is present diffusely throughout the cell. So this is multiple lines of evidence saying that these T cells are undergoing apoptosis and cell death. So interestingly to us, this actually was pretty strongly correlated with age in that all of the patients who were basically over 70 had fairly large percentages of these VDAC positive cells. There was a much greater distribution with some people with very high and very low levels, and then young people, it was fairly uncommon to have high levels of this. And this did, in fact, correlate with lymphopenia or low T cell count. So actually, the percentage of these cells, particularly when you look at the time point at which someone's actually already been seriously ill for a week, there's a really strong correlation between having high numbers of these VDAC positive cells and low peripheral lymphocyte counts. So apoptotic pathways being activated, maybe not super surprising that we then see small numbers of T cells as this process goes on for an extended period of time. Okay, so just to summarize the T cell work quickly, what we're seeing in severe, basically in patients who are at older age and have basically lower lymphocyte counts, is that there's upregulation of VDAC on the surface of the T cell, apoptotic pathways, cytochrome C is released. You can actually inhibit that in vitro with a pan-caspase inhibitor or an inhibitor of VDAC polymerization. And that there's no effect on healthy cells when you treat with these inhibitors, but in the acute COVID patients where we have this upregulation of VDAC and the release of cytochrome C, these caspase inhibitor or the inhibitor of VDAC oligomerization actually prevent T cell death in vitro. So these seem to be distinct features of T cells, particularly in older patients, and that, again, was associated with lymphopenia. So this is a metabolically novel subset of T cells in patients with acute COVID. They're distinct from every other of the acute and chronic and severe influenza diseases we studied. They increase with age, and again, they have these really irregular mitochondria, activation of cell death, cytochrome C in the cytoplasm, and apoptosis. And we can rescue those cells in culture by inhibiting the two pathways I mentioned. So that's the summary of the T cell side. I'm not going to go through the myeloid data in great detail, but just summarize what we saw, because again, this paper is published. But basically, I already talked about the T cell side, but on the myeloid cell side, we saw expansion of immunosuppressive myeloid-derived suppressor cells, and I'll talk a bit more about that. But MDSCs, myeloid-derived suppressor cells, were seen very closely correlated with disease severity, and again, having high hexakinase 2 VDAC, particularly on the granulocytic or neutrophilic PMN MDSCs, and high levels of CPT1A as on the monocytic MDSCs. And when we did single-cell RNA-seq, we found that those MMDSCs were associated with secretion of chemokines and alarmins and enhanced reactive oxygen signaling, and that having HLA-DR positive antigen presenting myeloid cells is associated with mild COVID-19 and recovery. So HLA-DR downregulation with myeloid-derived suppressor cells is actually associated with poor outcome. So I'm going to focus in looking at, again, the T cell and myeloid cell differences we found in these populations. I'm going to focus for just a moment on these neutrophilic myeloid-derived suppressor cells. And so this was actually, again, very strongly associated with having acute COVID. And again, not really present to this extent, even in flu, but certainly not present in healthy controls or people who recover. And so this population, again, is metabolically distinct as well, but even just the presence of these granulocytic or neutrophilic myeloid-derived suppressor cells was really enriched in patients with COVID. And so what are these cells? And really, honestly, they've been best studied in the setting of cancer. They're known to be important regulators of the immune response to cancer, and the lectin-type oxidized low-density lipoprotein, or LDL receptor, LOX1, has been associated with their suppressor function. So it's undetectable in healthy people, but present in as much as 15% of the peripheral of total neutrophils in cancer patients, and very enriched in tumor tissue are these cells expressing LOX1. So LOX1 have gene signatures. The LOX1 positive neutrophils have gene signatures and potent immune suppressive activity, and there's upregulation with ER stress and other biochemical characteristics that really say these are suppressor cells and not just normal neutrophils. So the question, this has really been characterized well in cancer, but is LOX1 in the setting of severe acute COVID really indicative of cells that are suppressive? And if so, what's driving their production? So we know that in healthy people that it's upregulated by ER stress, but is that what's happening in acute COVID? So first, I just want to clarify that what I'm largely talking about in this is when we're looking at peripheral blood of patients is the low density neutrophils, because our normal density neutrophils get thrown out with the red cell pellet. So when we look at PBMCs from people with severe COVID, we see a marked increase in these low density neutrophils in this white cell layer. And so how are those then characterized? So as I mentioned, we see this really big increase in low density neutrophils in people with acute COVID compared to healthy control. And there's evidence of these cells undergoing degranulation. So without going into great detail, these are two markers of degranulation, both highly upregulated on neutrophils in patients with severe COVID. Because again, all of these patients were hospitalized. So I should point out there's a range of severity, but all of them were sick enough to be hospitalized. Okay, so and again, as a marker, at least in well-defined in cancer patients of LOX1 having immunosuppressive effects, the neutrophils have much higher percentage of LOX1 on them. And they're also more immature. So quickly, I'm just going to say that we then said, is there real meaning to this cell population? We expanded the number of people we studied from that original report, and we looked across a range of severity. So on the left is the state of the patient, like I'm running around totally fine. I didn't even know I had COVID until I tested positive, symptomatic, but still out and about, all the way down to somebody who ends up dying of COVID. And we looked at people who had mild to moderate COVID, which are stage 4 or less, or really stage 3 or 4, and then people who had more severe COVID, and we compared them. And we made sure that patients were not vaccinated or treated with dexamethasone to modulate what we were looking at. Okay, and so again, we really see that, as has been shown in many clinical studies, that this is peripheral blood and not PBMCs, but the absolute neutrophil count is very strongly associated with severity of the disease. And then again, we see this significant increase in low density neutrophils as severity increases, and then LOX1 neutrophils in these patients. So if you look at the proportion of people who have more mild, but again, it's not the mildest, like stage 3 or 4 compared to severe COVID, if I aggregate the data, again, the low density neutrophils, LOX1 positive neutrophils, much more common. And I lose this significant difference. Immature neutrophils are more present in COVID, but it's not different based on severity, nor is CD63. But when I look at also program death ligand 1 and LOX1, and this has also been shown to be upregulated on myeloid-derived suppressor cells, I see, again, a significantly higher expression in the severe compared to the moderately ill COVID patients. Okay, and this really peaks early. So unlike the T cells where I said, lymphopenia seems to occur a week or so into the illness, basically early on we see the same things I talked about being significantly elevated PD-L1 on the MFI of PD-L1, as well as the number or the percentage of low-density lymph nodes low-density neutrophils expressing this is really high very early on in the people who have severe disease. Okay, so just to point out that we don't just find these cells in peripheral blood. When we look at the bronchoalveolar lavage fluid or BAL from patients, we had comparators, healthy controls who were undergoing procedures and whom we actually got some BAL fluid as well. Basically, the low-density neutrophils, as well as these same neutrophil populations, the granulocytic MDSCs, both as a percent of live cells and percentage of low-density neutrophils, are increased in the BAL compared to the peripheral blood, and also increased in patients with disease compared to healthy controls. And PD-L1 does not seem to be as different between the BAL fluid and peripheral blood. Okay, so importantly, when we do a random forest plot and say, what are the predictors of disease severity, the things that really come out, the absolute neutrophil count, but in addition, the LOX1 PD-L1 percentages of live cells and of neutrophils is actually, they're both extremely important features for predicting severe COVID. Okay, so what's going on here? Neutrophils can be activated by a variety of stimuli, really pictured here. But one of the questions we asked was, does the virus itself induce this? Are LOX1 positive neutrophils suppressive? They've been shown to be so in cancer, but can we just assume that because there's LOX1 on the surface, they've adopted a suppressive phenotype? We should not assume that. And if they are suppressive, what would the mechanisms of suppression be? And actually, what we showed is that the virus can directly induce LOX1 expression on the surface of neutrophils. So if you just mock treat cells, treat them with SARS-CoV-2 or Hepzagarden, which is actually a positive control, it's known to upregulate LOX1, and the cells all end up dying after the first hour here, so we don't have the control. But subsequently, you can see that very rapidly SARS-CoV-2 upregulates LOX1. So this is not some, transcriptional change. Within an hour, the vast majority of cells are expressing LOX1 and at high levels. So so basically this is showing this graphically over time. Basically using SARS-CoV-2, Wuhan 1, we find that there's rapid induction. It's not quite as rapid as Hepzagarden, but it's very rapid, and that the levels decline, but actually so do the cells. The cells actually do die as we go later in time. And I just want to point out again that, remember, we didn't really see these cells in influenza. And in contrast, we used H1N1, which was predominantly what these patients had as the source of infection. And it lacks the capability, even after 24 hours of stimulation, of upregulating LOX1. So this does seem to be a distinguishing feature between the two viruses that might explain the prevalence of this population in severe COVID-19, but not in severe influenza. Okay, and so now we get at the question of, are these cells becoming immunosuppressive? So as I mentioned, when you treat with SARS-CoV-2, there's massive upregulation of LOX1 very uniformly across multiple donors. There's also a reactive oxygen species release as measured by dihydrorhodamine 123, and then there's also NETosis taking place. So it does look like it's inducing neutrophils to initiate some of the suppressive pathways. And so why are these cells present at higher frequency in peripheral blood? Because among other things that the virus does, it actually increases the percentage of low-density neutrophils. So this is why we're picking them up in peripheral blood. And that actually, becoming a low-density neutrophil happens when neutrophils get activated. So this is another source of evidence that these neutrophils have become activated. And what we see is, when we look at different, so neutrophils have primary, secondary, and tertiary granules. There are three components of degranulation that can be, that basically distinguish which granules being induced. It doesn't look like the secondaries are doing anything, but the primary granules and tertiary granules are being induced to degranulate upon SARS-CoV-2 stimulation. Yes, these are in vitro studies trying to get at how we're seeing these cells directly ex vivo from patients with severe COVID. So I actually don't have the data shown here, but it does seemingly get in, but it does not productively replicate, meaning that if we actually cultured the neutrophils and cultured epithelial cells with, under conditions that are optimal for keeping both alive, and there was no evidence of productive replication of SARS-CoV-2 within the neutrophils, although we, of course, got it to replicate beautifully in productive infection in the epithelial cells. This was done in Andy Pekosh's lab by my grad student who ran back and forth between the labs. So that's a great question. So it does not seem that the virus is being produced, or it's not producing in any, not just infectious virus. We couldn't even find genomic material from the virus, a few hours after we washed everything off. So didn't show the data, but I have them if you'd like to see them with me. And it's basically not productively infecting neutrophils. So, and as suggested by the change in 1 hour and my saying this isn't a post-transcriptional change, what we essentially are doing is decreasing the intracellular, and this is just LOX1 labeled on two different fluorophores, you can basically see LOX1 levels go down inside the cell and go up on the surface. So we're really just, again, taking previously formed LOX1 and expressing it on the cell surface. Evidence of that, if we basically use this RAB inhibitor that inhibits degranulation, we find that we have less of LOX1 upregulation with SARS-CoV-2, and then again, less primary and tertiary granule degranulation. So basically, degranulation of neutrophils, for which we've shown evidence SARS-CoV-2 induces, can cause tissue damage, recruit immune cells, and mediate immune suppression. And there are a variety of ways in which degranulated neutrophils do this: reduction of proteases, reactive oxygen species, arginase 1, IDO, and PD-L1. And so we asked, what's happening here? And could this actually suppress T cells? Might this be a problem? We've already shown that in older people, we get lymphopenia. Now, are we actually also inducing cells that suppress T cell function? And the short answer is yes. So it turns out that if you treat these cells with SARS-CoV-2, just like we saw directly ex vivo, we have high levels of PD-L1 on the surface of these neutrophils. And so basically, there's a dose-dependent effect where the more virus, this is the multiplicity of infectivity of SARS-CoV-2, the more virus leads to more PD-L1 expression, which, as you know, can engage program death 1 and potentially inhibit T cell proliferation. And that, as has previously been the case, when we inhibit degranulation, that effect is lost. So we then ask the important question, okay, does this population actually suppress T cells? We can assess PD-1 proliferation. Here we're using program death 1 as a marker of activation because in the acute phase, PD-1 goes up when you activate a T cell, and we look at production of cytokines to ask, are these neutrophils actually suppressor cells and not just neutrophils? And the data basically are shown here of the T cells with the virus present on with neutrophils that have either been, the T cells present with neutrophils. There's a little bit of suppression if you just put in neutrophils, but that is shown here. This is, again, a positive control, which actually, while dapsogardin induces neutrophils to upregulate LOX1, it pretty potently suppresses T cell proliferation. But you can see that the virus itself can also, when the virus has stimulated the neutrophils, they begin to suppress T cell proliferation as well. And that's summarized here with varying ratios of neutrophils to T cells, showing that these neutrophils, when they've been exposed to SARS-CoV-2, actually inhibit T cell proliferation. And this T cell proliferation was induced by anti-CD3, anti-CD28, so not SARS-CoV-2 specific. Okay. And that basically this suppression, the level of suppression was correlated with the level of reactive oxygen species release, LOX1 upregulation, and PD-L1. So it looks like upregulation of all three of these is associated. I'm not showing you here, but we didn't see any effect of arginase 1. So while neutrophils can produce that, there was no relationship between arginase 1 production and suppressive effect, and the suppression seemed to be mediated by these. So then basically what happens to the cytokines made by these T cells? I'm sorry, I've lost the significance factors here, but you can see that while interferon gamma production is not really significantly altered, you see a particularly potent suppression of IL-2, not surprising given the absence of proliferation, IL-4, and TNF. So these neutrophils are suppressing T cell proliferation, T cell cytokine production, and presumably T cell activity. This can be inhibited by basically inhibiting degranulation. As I mentioned before, we actually get better proliferation of T cells when we inhibit degranulation. We also used a monoclonal antibody that's directed against PD-L1 to block PD-L1, PD-1 signaling. And actually that partially also reduced and I'm sorry, again, I've somehow lost my significance markers, but that also decreased the amount of suppression. So really what these data show is that the T cells are suppressed by the neutrophils when the neutrophils are able to degranulate and the neutrophils upregulate PD-L1. Okay. So in summary, SARS-CoV-2 drives neutrophil activation that results in functional suppressive signatures, PD-L1 and reactive oxygen species, that requires degranulation and PD-L1, but as I mentioned, but didn't show not arginase 1. And neutrophils being recruited to the lungs may be critical defense mechanisms in a variety of settings, but SARS-CoV-2 seems to drive neutrophils into a suppressive phenotype that we found in blood and lungs that seems to alter T cells' ability to respond to viral infection. So that combination in older patients of SARS-CoV-2 being associated with lymphopenia and these cells that suppress T cell proliferation and function may in part explain the severity of disease. Okay, so with this part, I have many people to thank, members of my lab. The neutrophil work was primarily driven by Lian Su, and the work on the other earlier part on metabolic signatures by Liz Thompson, but it was a whole team effort. So switching topics and wrapping up, I just want to talk a little bit about some of the work we've done in assessing responses to vaccination and not just severe infection. And these are basically on the axis here, compared to healthy people, a variety of risk factors for failure to respond to vaccination. And these are data following the original recommended course of 2 vaccines against SARS-CoV-2. And you can see that the most significant risk factor for failure, while reversed in many of these settings, was actually in solid organ transplantation, where the responses were low, and the percentage of people who seroconverted were lower than in these other groups, all of which are risk. And associated with this is an increased risk of breakthrough infections in solid organ transplant recipients, who are actually, unfortunately, also at higher risk for severe disease. So we basically, this was before everybody got a 3rd dose early on. We asked, would a 3rd dose benefit immunocompromised patients? We initiated 3rd doses of COVID vaccines in solid organ transplant recipients right after the 2-dose regimen was recommended, and asked, importantly, scientifically, in the people who failed to respond to a 2-dose regimen, if they respond to a 3rd dose, what would be different about the antibodies and memory B cells? And do we see anything that might explain that? And then we asked the question, are there specific immunomodulators or suppressive agents that are associated with VARI2 or succeeding in response to a 3rd dose? And what is the mechanism of response if we can get them over the finish line with a 3rd dose? Adding to that, our interest in that I've already outlined for metabolic profiling, is there anything metabolically different about the cells in responders versus non-responders? So again, going back to my chart, but now instead of looking at T cells and myeloid cells, which were the focus, we'll talk about B cells and how that affects B cell function. So first of all, we could label SARS-CoV-2 spike protein specific B cells by using 2 different fluorophores on labeled antigen, labeled spike protein, to identify with the double positives being specific for SARS-CoV-2 spike protein. And what you find is that, this is a pretty specific assay, in that here unvaccinated people not exposed to SARS-CoV-2 and class-switched and unswitched antibodies. And of course, you see in vaccinated healthy controls, robust induction of spike-specific class-switched and unswitched antibodies. And so we then asked what can we learn about the B cells in these individuals? But the first question was, is there anything clinically can find that explains response and non-response? And multiple studies have shown, in addition to ours, that basically you can make class-switched antibodies on profound immunosuppression, which honestly, it was a little bit surprising to us that people are on high-dose immunosuppressive agents, so they'll tolerate the transplanted organ they received, can actually do this, but there are some class differences and not a significant difference in IgM. And so we hypothesized that some of this would be based on the ability to mobilize T cell help, and we looked at T cells as well, but the focus is mostly on B cells. So they're skewed toward the unswitched phenotype. So I showed you the healthy control data. Now we're looking at solid organ transplant recipients, which I'll abbreviate SOTR, and they basically are skewed toward an unswitched phenotype with a significant difference in the percentage of class-switched antibodies. But again, in these very profoundly immunosuppressed patients, we saw that some of them with a 3rd dose of vaccine could actually go from undetectable antibodies against spike protein 2 detectable, while others of them really didn't respond to vaccination at all. So clinically, we asked, what might be different? Well, first of all, the people who responded well to a second dose, not surprisingly, tended to respond to a third, but there were some people who were non-responders, as mentioned, following even a third dose. So we asked, what are the clinical things that might explain this? And I'm not going to show the detail, but these solid organ transplant recipients had received kidneys, hearts, lungs, mostly kidneys, and honestly, there was no correlation with the organ transplanted. And we actually looked at whether they got Pfizer or Moderna, no difference with the vaccine received. But there was a profound difference based on the immunosuppressive regimen prescribed. So there are a variety of immunosuppressive agents used in patients to help them tolerate a transplanted organ. And what we found really to be relevant was mycophenolate use. So as shown here, basically it's a de novo pairing synthesis inhibitor. That is the documented mechanism of action of the drug. And we broke it down between responders, who are, again, shown in this teal color, and non-responders in red. And while there were some non-responders, there were some responders in the group of people who received mycophenolate mofetil, there were also the only people who didn't respond were on MMF. So you could respond on MMF, but there were no non-responders among the people who were not receiving MMF. Multiple clinical studies have shown MMF to be the problem in vaccination in this patient population. And so, really the chunk of the matter is here. There are a whole bunch of different compounds we've looked at, but it's really MMF that's the problem. So does that help us learn anything about what the mechanism is? As I mentioned, MMF and the active metabolite of it is mycophenolic acid, MPA, basically is, as I mentioned, a de novo pairing synthesis inhibitor, making it a potent inhibitor of T cell and B cell function. And so that is the known mechanism of action. And we actually looked at, again, metabolic roles. We actually subset the B cells based on whether they basically had expression of CPT1A, again, the rate-limiting enzyme and long-chain fatty acid synthesis, and found that the responders actually seemed to express higher MFI of CPT1A. So it looked like CPT1A or long-chain fatty acid metabolism was helpful in this. And when we actually asked what are the characteristics of cells that predict response to a third dose, we identified different clusters that were predictive of non-response or response, and basically then asked what is unique about those clusters of cells. And so specifically, rather than going through each of the clusters in great detail, I will just say that in the people who were responding, meaning upward arrow means higher in responders, we saw more of these alternative lineage CD11C positive B cells that, again, expressed a lot of CPT1A as associated with good responses to vaccination. So what are these cells? They go by a number of different names: alternative lineage, atypical B cells. They're associated with being older, with immune cell senescence, and they're more frequently seen in people who have conditions like HIV, malaria exposure, and autoimmune disorders. I will say that both systemic lupus erythematosus, or lupus, and rheumatoid arthritis are also treated with MMF, which may be relevant as well. So they actually seem to have the improved ability to present antigen to T cells, and there is an increased stability of the interaction between CD11C positive B cells and T cells. They are, in fact, part of a normal response to vaccination, and we find them in patients who've been vaccinated against malaria. So this is the Plasmodium falciparum circumsporoid protein. And so you basically see them in malaria naive individuals, and they are mobilized in those vaccines. But we then asked, okay, well, so is there something different about the response to vaccination? And for starters, we found that the CD11C frequency was a bit higher in the people who received solid organ transplantation, but I can tell you that they're actually present in healthy donors as well, so it didn't seem in the end to be about frequency. And we thought it was more related to CPT1A expression. And there is more CPT1A expression, and high-dose MMF is associated with both lower expression of CD11C and CPT1A. So we perform seahorse assays, and these are basically assays in which you can assess the oxygen consumption rate or mitochondrial activity, as well as the rates of glycolysis. And without going into great detail here, we can show that we use doses of MPA, which is the active metabolite of MMF, and it had the effect expected in that it definitely inhibited T cell and B cell proliferation. So it does work in the way that it says it works, at least in that it is an inhibitor of T cell and B cell proliferation. But we found, interestingly, in individuals who received, who were patients, so this is patient data, taking cells from people who were either being treated without MMF or had low-dose MMF or high-dose MMF, and comparing to healthy controls, we saw some baseline upregulation of respiratory function, of oxygen consumption rate at baseline that was negated in the patients in high dose. So surprisingly, they actually looked like they had higher rates compared to healthy controls if they weren't on MMF at all or they're on low dose, but that high dose MMF basically decreased this oxygen consumption rate at baseline. And so that's probably the thing to focus on. And in vitro, we could actually mimic this by basically showing that compared to untreated individuals, low-dose MPA brought up oxygen consumption rate, and high-dose MPA started to bring it back down towards healthy controls. So could we actually prove this was dependent on endogenous fatty acid oxidation? And I should say that, this is not isolating CD11C positive B cells, but in general in the PBMCs. And the thought was that low-dose MPA could actually help the cells to take up endogenous lipid droplets and increase OCR without exogenous substrates. And that was borne out by the seahorse data. So the question is high-dose MPA inhibiting fatty acid oxidation required for this process to take place? Why is high-dose MPA reversing this? So we actually did this in many patients, and I'm just showing you one example, but and basically MPA treatment actually induces accumulation. This is showing with oil red O, but we use multiple ways to show that MPA treatment actually is associated with lipid droplet formation in B cells, and that actually is associated with their inability to use exogenously added substrates and process them normally as a source of fuel. So to summarize the multiple things we tried, including adding a variety of different sources of fuel to these cells, you can see that the cells, when they were treated with MPA, basically had significant differences in their ability to respond to a variety of exogenous substrates. So high-dose MPA, in contrast to low-dose MPA, does not allow the cells to use lipids as a source of fuel, but instead results in lipid droplet formation and toxicity to the cells that ultimately decreases their ability to use lipids as a source of fuel with decreased oxidative phosphorylation. So what are the impacts of this? Are these cells basically able to function normally? We looked at proliferative capacity and differentiation into plasma cells of B cells, either the CD11C negative cells or the CD11C positive cells, untreated, given low-dose MPA or high-dose MPA, again, the active metabolite of MMF. And we looked at their ability to proliferate with and without etamoxir, which inhibits fatty acid oxidation. But I'm not going to talk about that because it has, I'm really going to focus on just proliferation. And so if you look at the CD11 negative B cells and you basically don't treat them, when you find that at baseline, the CD11C positive B cells stimulated to differentiate actually differentiate a bit more into CD38 positive plasmablasts. And again, they lose cell trace violet when they differentiate. And that is preserved in low-dose MPA, but you begin to see an advantage of the CD11C positive B cells in low-dose MPA with enhanced proliferation. In contrast, in high-dose MPA, neither the CD11C positive nor the CD11C negative, neither B cell population can proliferate and differentiate into plasmablasts. So low-dose MPA doesn't seem to adversely affect this ability, and the high-dose MPA, which was most closely associated with clinical failure to respond to vaccination, does result in impaired proliferation of B cells. So in summary, we were surprised to find that despite immunosuppression, 72% of solid organ transplant recipients could mount spike-specific antibody responses after a third dose of vaccination, that those that failed to respond to a third dose uniformly received mycophenolate, that low-dose mycophenolate mofetil induces fatty acid oxidation of lipid droplets, which results in increased basal mitochondrial metabolism. But in contrast, high-dose mycophenolate inhibits rather profoundly fatty acid oxidation mitochondrial responses to exogenous substrates we gave in vitro. So MMF inhibits some of the unique metabolic requirements that B cells have as a pathway that can allow solid organ transplant recipients to respond to vaccination. And this has not actually ever been described as the mechanism of mycophenolate mofetil, because as I mentioned, it's really been studied and investigated and marketed as an inhibitor of de novo purine synthesis. But in fact, high-dose MMF really profoundly shuts down the ability of B cells to use fatty acid oxidation and make responses to vaccine. I didn't show these data, but we did RNA-seq on cells from patients being treated with MMF compared to those who are not. And the RNA-seq data show that these B cell populations have radically altered cholesterol homeostasis gene expression. And statin use is associated, statins which lower lipid levels are associated with better antibody responses in people on MMF, but there's not a difference in the patients on statins if they're not also receiving MMF and their ability to respond to vaccination. This potentially offers a manipulable pathway to enhance responses to vaccination. And with that, this part of the project was conducted, again, with Elizabeth Thompson, who is just wrapping up and going on to the faculty now at Hopkins, and Justin Bailey, a B-cell expert at Hopkins, along with a whole host of people with advice along the way from Erica and Ed Pierce, who are world-renowned experts in immune cell metabolism. And in all of my collaborations on the antibody side involve the work of Sabra Klein and Andy Peckosh helps us with a lot of our assays in which we are adding virus and seeing what happens. So